Good morning, Wolverines! And now it's time for your essential questions. Number one, what is gene linkage? Number two, how does linkage relate to gene inheritance and segregation? And number three, what are autosomal chromosomes and how is this different from sex chromosomes with respect to inheritance patterns? Lastly, four, can you determine if a trait is recessive or dominant by looking at pedigrees? If so, how? So there are two main focuses for today's notes. We want to teach you what linked genes are, and then to give you a better idea of the impact of that, we're going to start studying pedigrees. And so pedigrees are essentially like a modified version of a family tree where you're examining the impact of a particular trait over generations. So you can see where you inherited a specific trait. And you can start thinking about what that means in terms of allele combinations and genetics. So let's start out, however, with looking at linked genes. So linked genes are genes that are found on the same chromosome. All right. They're inherited together as a unit and do not undergo independent assortment, which is one of the things that we talked a lot about with Gregor Mendel, is that genes were inherited separately. And while that's true for a lot of genes, if they're on the same chromosome, they're not inherited separately. So if we take a look at this homologous pair, you can see that we have gene A and we have gene B. Well, notice that if you get gene A, you're going to inherit most cases gene B at the same time. Okay, so if you have a dominant A in this example, you're going to get a recessive B. If you have a recessive A, you're going to get dominant B. Now, this is going to change our inheritance patterns. You're not going to always have a 9 to 3 to 3 to 1 that we saw in our activity yesterday with a dihybrid cross because you're not really going to inherit big A with big B because they're physically connected together, big A to little v. Okay. The only time that this doesn't happen is as we're going to form those gametes as we go through meiosis, if you have a crossover mutation. If you have a crossover mutation, you're not necessarily going to get the exact same combination that you see here, where you have big A with little b and little a with big B. You could get other combinations depending on where the crossover occurs and how the body does or doesn't correct it. So let's start by taking a look at linked genes. Now, there is a typical way that we write linked genes when we're going to go ahead and show them in terms of inheritance, and that's with this solid line between the pairs. Okay, so in parent one, you can see here that we're going to go ahead and deal with dominant A and dominant B being physically linked because they're together above the little bar. The other option in terms of the homologous pair is lowercase a and lowercase b because they are linked below the bar. So the bar represents the separation of the two homologous chromosomes. And you can see exactly that here when we go ahead and take a look at this. In terms of the separation, if you get big A, you get big B. If you get little a, you get little b because they're physically connected to each other. And what's the only thing that's going to separate them apart? Good. So it's a crossover mutation. Okay. So linked genes like this tend to be inherited together. And when we go ahead and start looking at this means that there are fewer combinations of alleles in the offspring compared to genes that do separate because they're found on different chromosomes. So in describing linkage, the appropriate notation shows a horizontal line where it's just separating the two linked groups as you see here. Okay. All right. So let's give you an example so you can kind of see how this plays out in real life. All right. Drosophila melanogaster, or the common fruit fly, is a typical experimental animal that we go ahead and utilize when we're doing genetic tests and genetic experimentation. And the reason for that is they have a pretty small genome, as far as animals are concerned, and we can go ahead and get them to reproduce really quickly so we can see a whole lot of generations in a rather short period of time. Now, in this particular example, we're going to be examining the genes for wing shape, whether it's straight or curled, and for body color, whether it's gray or ebony. And they're located on the same chromosome and therefore inherited together. So if you take a look at these two flies, you have your wild type female and your mutant male. Now, if we look at the phenotypes for each of these, you can see that the wild type female has straight wings and a gray body, but the mutant male has curled wings and an ebony body. So if we go ahead and take a look at these particular traits, you can see that the dominant here of CU and EB, curved wings, and ebony, and it turns out that the terminology here is kind of the reverse of what we've been doing. They use the dominant CU and EB to represent straight wing and gray body because while they started the investigation by examining the mutant version, which is where the letters came from, they found out that both of these traits are actually recessive. And so you see in the mutant male the lowercase or recessive CU and lowercase or recessive EB, and they're homologous for it. So the only thing they can show is the recessive trait, which is what you see in the mutant male. On the other end, the wild type female does not exhibit those traits because it has the capital CU for straight wing and the capital EB for gray body, and those are dominant over the recessive curled wing and ebony body genes. So how would you write it? Think about the previous slide. Hopefully you were able to remember that we write this in linkage notation by using that horizontal bar to go ahead and show which two versions are connected to each other. CU to BEB, dominant with dominant, or CU recessive with EB recessive. Okay. All right, so that gives you a basic idea of what a linked gene is, and we'll go ahead and talk about these more as we go through pedigree analysis, and it'll come up as we keep going through genetics. So let's go ahead and move on and start talking a little bit about pedigrees. Now, the first thing you need to know about pedigrees is that we have some key symbols that we're just going to have to go ahead and learn. In a pedigree, a circle is always going to represent a female, and a square is always going to represent a male. If you do not know the sex of an individual because they are in your extended family for some reason, or your family doesn't talk about it for whatever reason, these would be represented with a diamond shape, as you can see here. If you have an individual who died at infancy, we put a solid smaller dot, and this is because we don't really know all their traits because their physical characteristics usually develop in the first 2 years. Things like eye color, for example. Every baby starts off with blue until the pigments kick in and start showing whatever particular color they're going to manifest. An affected individual is somebody who has the trait. That doesn't necessarily mean it's a bad trait. I think we often associate the word affected as meaning with a disease or some sort of illness, but affected simply means that we have a particular trait that we're examining. And so then that's typically going to be shaded in or some darker color versus the normal, which is going to be non-shaded in or some lighter color. Now, some of you may be able to go ahead and think about families that have twins. But before we go ahead and start thinking about how to express this, we have to realize that there are two different types of twins from a genetic standpoint. We have our identical twins and our non-identical twins. Now, you'll notice in this instance, non-identical twins start at the same point because they're born at basically the same time, but they have their own separate lines coming off the horizontal. This is important because non-identical twins come from their own separate eggs. This is why non-identical twins can have male and female at the same time because they do not have the same DNA. This is not the same when we start looking at identical twins or even triplets or whatever number you may be looking at. Identical twins start out as one egg that then splits and forms into two separate eggs as they develop. Okay, so identical twins do start out with exactly the same genetic material. Now, we can talk about mutations and how they accumulate over time and realize that by the time they're adults, there are probably some variations in their DNA. But at the base level, when that egg is first developing, they start out as the same egg, so they have the same DNA. Once we start going ahead and creating this, we then need to kind of think about how we can signify inheritance. Now, when we start looking at what's known as a carrier, we're going to go ahead and represent them either with a dot like this, or if I can go ahead and draw this really quickly. Let's see here. If I go ahead and make a little circle around here, by going ahead and shading in half of it. Okay, so we can go ahead and draw a line down the center, and then we can go ahead and color in the other side. All right. So either one of these is perfectly acceptable for when we do a carrier. Now, in many cases, I've more commonly seen the half shaded, like I'm drawing right now, versus the dot in the center, which you go ahead and see here. But either one is considered acceptable. And by a carrier, what we mean is an individual who has the trait that you're trying to follow, but doesn't show it. Okay, so this is going to be a heterozygous individual, and you're typically in these types of cases looking at a recessive trait. So the dominant trait is hiding or covering up the recessive, but they do have it to be able to pass it on to future offspring. Then we go ahead and start looking at generations. Now, this could be your grandparents, your parents, your generation, your nieces, nephews. Hopefully, none of you have kids yet. And we represent these with what are called Roman numerals. And so you can see the I. So these are how we signify generation, is using Roman numerals. Then we try, and we'll talk more about this when we get examples later on, to go ahead and number the children in birth order. And for this, we typically go from left to right, and we go ahead and use Arabic numerals, 1, 2, 3, as you see here. So individuals are designated by a combination of their generation and their order of birth. So you would have something like Roman numeral 2-2. So that would be generation 2, individual number 2. And that's how we go ahead and signify or designate a particular individual. All right. So here's the sample pedigree. Oh, I'll go ahead and take off my carrier here. And if we take a look at this, this is obviously already missing the generations. We could go ahead and label this if we wanted to. Let's see. This generation is going to be generation 1. This is going to be generation 2. And this is going to be generation 3. Okay, notice that I did this with Roman numerals. And then we can go ahead and label the birth order as well. Typically, you want to go ahead birth order, oldest on the left, youngest on the right. We can go ahead and label this with a 1, this one with a 2. We can go down here. We'll start over because we're doing a new generation. Okay, there's 1. There's 2, and so on and so forth. I won't continue to do this, but you get the idea. Now, a horizontal line for the longest time represented a marriage. Now, we all know at this point in time that there are plenty of children who are born outside of a marriage. So this typically represents now what we would consider to be a union that goes ahead and produces offspring. So in this particular instance, you can go ahead and see up here at the top that we have this female, it's a circle, so it's a female, and had children with this square, this male. The square is not colored in, so this is an unaffected male, and this is an affected female. They have children represented by this bar going down and then this bar going across. And so stop for a second. How many children did they have? Take a minute, think about it. Okay, How many did you come up with? Well, hopefully you came up with 4, because the only ones that are actually their children are the ones that are connected to this horizontal line. So we have 1, 2, 3, and 4. They had 2 daughters and 2 sons. Now, yes, there's individual 2-2 here, and you've got this female here and this female here, but those are individuals who married into the family or had babies with people from the family. And so we don't know necessarily as much about their genetic history. Again, individual 2-1, which is this individual right here, is half shaded, so this is going to be a carrier for whatever trait they're looking at. This male is affected with whatever the trait is, and then you can see these 2 females here, as well as this male here, are carrying at least that particular allele. This male, like this male, is affected, and these 2 females, like these females, are carriers for that particular trait. Okay. All right. So here's another example. And the new term, this is a vocab word that you'll want to make sure that you understand, is prepositus. So when we start identifying a pedigree, not a Punnett square, the person who started the investigation is known as the prepositus. And so you can see down here, it's typically signified with some sort of arrow. And this represents this was the individual who started the investigation that produced this particular pedigree. All right. In this particular instance, we have square. What's that represent again? Good, male. And then we've got a circle, which has to represent, good, female. The male in this instance is affected with the trait, and the female in this instance is a carrier, but not affected by the trait. If you take a look in the next generation, we have 4 children again, 1, 1, 1, 3, 1, 4, and 1, 5. And when you take a look at 1, 3, and 1, 4, hopefully we recognize that they are what? Good. Hopefully you said identical twins, because the bar comes down from the line here first before going ahead and splitting off into the 2 offspring. When we go to generation 3, we look at individual 3, 1, and we see a diamond. Do you remember what the diamond represents? Good. So this means unknown gender. So we don't know if it's male or female for various different reasons. Then we look at individuals 3, 3 and 3, 4, and we recognize that they are, hopefully you said non-identical twins. Notice that they're different genders. The bars go up to the same point of the line, but they do not come down from the line first before splitting. And then if we look at individual 3, 6, okay, that solid dot represents what? That's right, that they died in infancy. Okay, so those are key things that we need to kind of look for as we go ahead and start identifying pedigree charts to go ahead and try and interpret them. All right, so let's go ahead and get started. Now, before we start going too crazy, I do want to talk about 2 key terms. Okay, when we go ahead and start looking at these terms, let's pick a color. It's going to be easy to read but not disappear. We have autosomal. Okay, now autosomal traits are what we would consider on body chromosomes. Okay, autosomal are body chromosomes. And this is to be differentiated from what we would call sex chromosomes. These are going to be the X and Y chromosomes. And these are the ones that determine the gender of the offspring. Okay. So when you go ahead and take a look at a karyotype, and remember a karyotype is all of your chromosomes, okay, we see these little X's. That's what we're used to seeing. Now in a karyotype, they're not going to be in these X's because these are actually duplicated chromosomes that are waiting to go ahead and divide. So a karyotype is going to have a little squiggly line and another little squiggly line. And let's say that this is for, go ahead, and chromosome 1. You'll see the same sort of thing again, and this time it'll be for chromosome 2, and so on and so forth. So when we start looking at these pairs of chromosomes, remember we have a total of 46 chromosomes, 2 sets of 23. Chromosomes 1 through 22 are your autosomes. Okay, so 1 through 22 are your autosomes. The last two, the X and the Y, are your sex chromosomes. And I only point that out now so that we can make sure that we understand this basic information for as we go ahead and get ready at understanding pedigrees. So let's start out with autosomal recessive traits. As we just mentioned, autosomal traits are typically going to be found on one of the first 22 chromosomes, and both men and women have the same number of genes. The recessive trait, remember, is the one that can get hidden or seem to disappear behind the dominant trait. On the next page, I'll go ahead and show you a pedigree for albinism. Now, albinism is the lack of pigment in your hair, skin, and eyes. It's inherited as an autosomal recessive trait. The trait is not sex-linked and is shown in both males and females equally. The affected female in the third generation has phenotypically normal parents. Let's take a look. So here is your pedigree. Again, notice the traditional traits we've been talking about. Okay. When we go ahead and take a look at this, you've got generation 1, generation 2, and generation 3. Then you're numbered based on birth order, going across 1, 2, 3, 4, through 5, and through 4 again. The horizontal lines at the top represent a union that produced offspring. The horizontal lines here represent that these are offspring of this union. If you see the dot in the middle of everybody in the second generation, remember this represents carrier, somebody who has the gene or trait but doesn't show it because it's a recessive and it's hiding behind the dominant allele. In generation 1, individual 1-2 is an affected female, and individual 1-3 is an affected male. Since they're affective, we can tell this is recessive because the only thing they had to pass on was the affected trait, which is why every individual in generation 2 is a carrier. If we take a look at individuals 2-3 and 2-4, these are the parents from the two separate families. They have children and they are both carriers. When we look at generation 3-2, we notice that 3-2 has whatever the issue is, in this case albinism, and they received it from their two parents, 2-3 and 2-4, who did not have albinism but were both carriers for that particular trait. So 3-2 is an albino girl whose paternal grandmother and maternal grandfather were also albino. Her other relatives are all phenotypically normal, but genetically, in terms of their genotype, they carry the recessive allele for albinism. All right, let's give you another example. In this case, we're going to look at an autosomal dominant trait. And this particular autosomal dominant trait is for woolly hair syndrome. Yes, that's a real thing. The inheritance of autosomal dominant traits is a little different than autosomal recessive. In order to get a recessive trait, you need 2 copies. But in order to get a dominant trait, you need only 1. So unlike our first example where the trait seems to skip a generation, in an autosomal dominant trait, it will be visible or present in every generation. If a large number of families are examined, about equal numbers of males and females are affected by an autosomal dominant trait, because remember, autosomal is one that's found on the body chromosomes 1-22, not found on the sex chromosomes of X and Y. So here's our Punnett square. When we go ahead and take a look at this, notice that female 1-2 is heterozygous for the woolly hair allele. We can tell that because, yes, she has woolly hair, so she's got to at least have one copy. But we know she's heterozygous because if you look at individuals 2-5, 2-6, and 2-7, they do not have woolly hair syndrome, which means mom had to be able to give them a non-affected allele. Okay? Individuals 2-1, 2-2, and 2-3 got woolly hair syndrome from their mother. Now, when you look at the offspring of 2-3 and 2-4, notice that when you get to the bottom, 3-1 and 3-2 do not have woolly hair syndrome, so we know that individual 2-3 is heterozygous. You can also tell that by looking up at generation 1 and noticing that they have an unaffected father who can only give them the recessive unaffected trait. Okay, now why does this matter? Because when we get to sex chromosomes, not everything is created equally. If you take a look at this photograph, this is a scanning electron microscope image of the X and Y chromosomes. Notice the Y is a dinky little thing. All right. There is a lot of information missing from the Y chromosome. So inheritance of traits that are found on the X chromosome is different for males because they often have just one copy and get whatever's there, whether it's dominant or recessive. This is because most sex-linked genes are found on the X chromosome, what we call X linkage, and do not have a corresponding match on the smaller Y male chromosome. There are a few cases where you have a phenotypic trait that is determined by an allele on the Y chromosome, but because the Y is so small, it's not many. Okay? It just isn't. So, fewer Y-linked traits, more X-linked traits, and therefore Y-linked diseases are far less common. Okay, so why did this happen? And the answer is, historically speaking, we don't know for sure. But if you think about something like a crossover that we talked about before, that genetic recombination, remember that your body repairs it by cutting it off and trying to reconnect it back together. Now, what if you lost half of that information? Now, you would have a chromosome missing a whole block of information like what you have here. And it could be passed on still because the individual had another copy that compensated for their lack thereof. Okay. Let's take a little bit of a closer look at the inheritance pattern of sex-linked traits. In this particular example, we're going to go ahead and show you a father who does not have any of the trait that we're following. He's got a healthy X chromosome and a Y that's too short to go ahead and have the disease on it. Now, fathers pass sex-linked alleles onto their daughters, but not to their sons, because again, most of them are located on the X chromosome. Mothers, however, can pass sex-linked alleles to both sons and daughters because all they have is an X. In females, sex-linked recessive traits will be expressed only if you're homozygous for it, but in males, receiving the recessive allele in just one chromosome will be expressed because you only have one. So let's say we have a father and he has 4 kids. He's going to go ahead and have 2 boys and 2 girls. All right. And he's worried about the fact that his wife has a particular disease. So let's go ahead and essentially do a Punnett square for these individuals. Father with X unaffected Y and mother with X unaffected X affected. All right. She's a carrier of the disease that we're looking at. So what do you think for the kids? Well, let's go ahead and start by taking a look at dad. We know that dad gave all of the boys what chromosome? That's right, A Y. And we know that he gave all the girls what chromosome? That's right, his X. And remember, his X is not affected by the disease. Now let's go ahead, oops, jump too far, and take a look at moms. When we look at mom's got one unaffected X and one affected X. If we start with the two kids at the beginning that did not get the affected X but got the normal X, we can see that the little boy is going to get an unaffected X from mom and a Y, which doesn't carry the information, from dad. He does not have the disease. He's also not a carrier. We go ahead and take a look at the little girl here. She's also going to get the unaffected X from mom, and she gets the unaffected X from dad. So she is homozygous unaffected and is just fine. Next, we're going to go ahead and take a look at the other daughter. But this time, she got an affected X from mom. Okay? So affected X from mom and unaffected X from dad is going to produce, not an affected girl, but a carrier girl. So this particular daughter has the trait but doesn't show it. So she still looks phenotypically or behaves phenotypically fine. And what about the last boy? He gets an affected X from mom. What does he get from dad? That's right. He gets the little dinky Y, which is missing all this information, and he does have the disease. So this is an important thing to kind of keep in mind. This is how things like hemophilia get passed on. What is hemophilia, you ask? It's an X-linked disorder in which blood clotting time is prolonged. Okay? It's famous because there was a whole line of people in many of the European royal families, because there was so much inbreeding, that had hemophilia. If you were in wartime and you got hit by a sword or something like that, it was very hard to stop the bleeding because you didn't have enough clotting factors. Nowadays, things aren't as big a deal because we can manufacture clotting factors, and you can have an individual actually take those clotting factors to stop the bleeding. Okay? So this works best when we go ahead and start looking at carriers. So women who are heterozygotes are carriers for the recessive allele, but do not have hemophilia themselves. However, they can pass that allele on to their sons, because sons, remember, will get a Y from their dad that doesn't impact them. And then they can express the recessive allele and have hemophilia. Now, the pedigree I have for you here is one from Queen Victoria. Now, she was famously a carrier of the allele for hemophilia. In fact, she ended up passing it on to one of her sons and through her daughters to the royal families of Prussia, Russia, and Spain. If you take a look at this, you can see Queen Victoria right here. And she's a carrier but does not have the disease herself. She's not a hemophiliac. And there's no family history of her husband, the king, having the trait. But then she has three children, two daughters and a son. Her first daughter didn't have any offspring, so we can't say for sure what her makeup was. But her son was a hemophiliac. And her daughter, we know, was a carrier because her grandson, individual 3-4, was also a hemophiliac. And you're dealing with a family on the other side, individuals 1-3, 1-4, 2-4, 2-5, and 2-6, who have no family history of hemophilia. Notice that there are no affected females in this instance, because affected females would have to have two copies of the hemophilia X, one from mom and one from dad. And the unaffected males here are not going to be able to pass that on. Okay. There is one more situation we should talk about, which is X-linked dominant traits. Now, these are, just like we saw when we were looking at autosomal dominant, going to be more common. But that's because you only need one copy to get the disease, rather than 2 copies or 1 copy with no match. Okay. Now, since females have 2 X's, they are more likely to get X-linked dominant traits, whereas X-linked recessive diseases are much less likely in females because they have another X that can compensate. Now, in this particular instance, we're looking at something known as rickets. For this particular instance, there's a rare pattern of inheritance where all the daughters of affected males will be affected, and more females than males will show the trait. An affected male must always have an affected mother because it's dominant and dad got his one good X from his parents. And so that's how he gets rickets. So let's go ahead and take a look at a pedigree going ahead and showing this. The inheritance of a rare form of rickets follows this inheritance pattern you see below. Notice that because it's dominant, it's in every generation. So some key things to look for. If it's in every generation, it's probably a dominant trait. If it skips a generation and you end up with carriers, it's probably a recessive trait. If it's equally found in men and women, it's probably autosomal. But if you have more men or more women, it's going to be likely to be sex-linked. All right. Now, if it's sex-linked dominant, you'll see it in more women. If it's sex-linked recessive, you'll see it more in men. In this particular example, the male 1-2 is affected with rickets, and all of his daughters, 2-2, 2-3, and 2-4, have rickets because they got the bad X from dad. The boy, however, 2-1, got a Y from dad, which doesn't carry the information, and got a good X from mom to make him unaffected. And even when individual 2-5 marries into the family and has children with 2-4, you'll notice that 2 of the 4 children have rickets because mom is the one affected and can pass that X on to any child, male or female. Okay. Now, you'll know that mom is not homozygous for it because individuals 3-1 and individuals 3-4 don't have rickets. Which brings me up to an interesting example. And this is what's known as bar bodies. In some instances, certain X chromosomes will be turned off. The chromatin that's still inside there gets kind of wound up densely and tucked off to the side and not used for expression in that particular cell. All right, this can happen in the nucleus of any non-dividing somatic cell. The chromosome becomes inactivated and you don't see whatever genes are on it that it would normally express. Now, random X inactivation is responsible for what we call tortoiseshell in cats. Calico is based on the same thing. You see patches where it's more than just 1 or 2 colors. That third color comes from this extra chromosome, and this is typically found on the X. And you may know somebody who has a calico cat. Any of you know somebody who has a calico cat? All right, good. So ask yourself, is it a male or is it a female? Now, based on normal genetics, it should only be female calicos because they have 2 X chromosomes to go ahead and turn one off and get the traits from the other one. But we also have instances where we go ahead and see it go the other way. All right. So the chromosome is inactivated, and what you get expressed is the one that's turned on, not the one that's turned off. So the only way we can end up with a male calico, because you have to have 2 X's to go ahead and do that, or more, I guess, is if they are a Klinefelter cat, meaning that their sexual chromosomes are XXY. And we'll talk more about that as we go through our activity for today. All right. But the presence of these bar bodies is very important in viable human aneuploidies. And that's what we're going to talk about. Remember, those are those mutations that are caused by a non-disjunction during meiosis 1 or meiosis 2. Now, in this cat, we have the tortoise shell patterning because we have black and yellow as two different fur colors. Black is dominant and yellow is recessive allele. So if we go ahead and look at this particular instance where we have that kind of little brownish yellow color, okay, this is an area where we have an active X that has the Y allele, and we have an inactive X with B, which has the dominant allele. So it's not going to show the dominant black. In those areas where we do see the black is turned on, but the Y for yellow, or the kind of tannish color, is turned off. And so we have both of those colors, and then if they're both expressed and nobody's turned off, you end up with the white. All right. So genes on one or the other sex chromosomes produce inheritance patterns that are different from what we would see with autosomes. They also show different results with a reciprocal cross. So what's a reciprocal cross? A reciprocal cross means that you are going ahead and doing the same breeding combination, but you traded in the genders. So whether it's the male or the female that has it. Okay. So when we go ahead and take a look at this, a reciprocal cross would mean having a colorblind female crossed with a normal male, and likewise having a normal female crossed with a colorblind male. If the results of these two crosses are the same, the trait is autosomal. If they are different, then what we end up finding out is that it's not set up that way. Okay? So it's not going to be autosomal, and it's going to end up being sex-linked instead. Okay? All right, couple quick reminders in terms of autosomal versus sex-linked. Remember that if it is an autosomal trait, all individuals carry two alleles for each gene, and dominance operates normally in both males and females. If we have a reciprocal cross, the results would be the same. Again, remember what a reciprocal cross is. And alleles pass equally on to male and female offspring. However, with sex-linked genes, these inheritance patterns do not hold true. Males carry only one allele of each gene. They're what we call hemizygous. Dominance operates in females only because remember, females have two copies versus the male only having one. And reciprocal crosses are going to produce different results. So how that gets passed on to future generations is not the same. You typically will end up having a little bit of a crisscross pattern. Father passes it on to daughter, daughter passes it on to grandson, and so on and so forth. All right, guys, so I hope that helps you understand autosomal and sex-linked genes. If you need help, please come in to class or to office hours to go ahead and get assistance with this. I do feel like this is one of the harder things to learn when we go through genetics, and we want to make sure that you're ready for when we get to our quiz. All right, guys, have a great rest of your day. I hope you're staying safe and healthy, and we'll talk to you soon. Take care. Bye-bye.